about how the proteins work in virus assembly, budding, scission, all the things. Yeah. All right, so we have modified and we have transported our proteins. Good. That's a good start. Now, the nucleoprotein, you can break down into two domains. There's an amino terminal domain, which they call the N terminal domain, or NTD. There's a carboxyl terminal domain called the CTD, and there's a thing called the N3 domain that's right at the tail. N3 domain is the one part of the nucleoprotein that's going to interact directly with M. The C terminal domain is very important for both binding to genomes, like specific packaging, and also for dimerization. So the two C terminal domains are going to come together. Here you can see them down here. The two CTDs of these two monomers are going to come together and make the N dimer. There we go. The SR-rich region is right in the center, and here you can see one copy, here's the other. These two molecules are kind of twisted around each other like that. And those together form like the unit of N as it will function for the virus. These units of N are going to be wrapped up with RNA, and we don't have a good crystal structure yet of RNA on these units of N, but I've got to think someday that's going to come, because that would be very nice to see. And this is an M protein above us. And then those units of N, that's what each of these blobs are actually going to arrange themselves in a kind of a helix arrangement. It's not a very good helix. It breaks, and so instead of being a long sausage, it ends up being a bunch of little sausage links with kind of random bits between. Each of these is not identical to the others. They can have different numbers of N subunits in there, and it all seems to work. But it kind of looks like a basket of spider eggs, honestly. In a nest, if you like. Yeah, if you're more a bird person. All right, here are the exciting interactions that are actually going to control packaging and assembly, because those are both dependent on M and N proteins, or strongly influenced by M and N proteins. Yeah, but at different parts. So, the red interactions here are known assembly interactions. So if you mess with those, you will block the M and N from interacting with each other efficiently. And you see those are both D's, so those are going to be acidic, negatively charged. This one's an R, so it's going to be basic. It's going to be positively charged at like normal pH. And so you got a positive negative interaction, almost an ionic interaction, that can exist between these things. So they could even form a little bond if they wanted to, or they could just interact via hydrogen bonds. All right, all the rest of these things. So there's 4 acidic residues and 1 basic residue. These all seem to be important for a specific packaging event. So packaging is going to involve a packaging signal. Now we know where the packaging signal is on some coronaviruses. This is mouse hepatitis virus over at the edge, and it consists of a long stem loop that's got 4 of these repeating units on it. And the repeating units seem to be the thing that the nucleoprotein is binding. So here's a dimer of nucleoprotein. We've got the one face, and then if you flip it over, you look at the other side of it, you can see it right there. You see there's a ring of these lysine residues that go around the edge. And if you interfere with any of those lysine residues, then packaging of genome but not subgenomic RNAs goes away, and this virus starts inefficiently packaging every viral RNA that it can get a hold of. So it's still dependent on what's interacting with the nucleoprotein, but nucleoprotein interacts with subgenomic RNAs and genomic RNAs, and now the packaging interaction can't tell the difference. So there is no use for the packaging signal unless all of these little lysines in this little ring can hold it. And so you can just imagine this packaging signal wrapping around with maybe one of these repeats up here, maybe one over here, maybe one down here, and maybe the 4th repeat there. And if you don't get all 4 repeats attached at just the right spot and just the right way, then it's not a packaging event. It can be non-specific RNA binding, but not packaging. And then these amino acids are also going to be important. These are in this C-terminal domain. all these amino acids. These are in the N3 domain, which is right at the tail. Yeah. All right, the packaging signal, where we know it in mouse hepatitis virus, is located in a loop between two domains of NSP15. Here's domain 1, domain 2, and domain 3, and it's between domain 2 and 3 right there. And just makes a little loop, because this is a thing that has a function as RNA. It's a packaging signal, but also it's in an area that's translated, and so it's part of a gene, and so it has to not interfere with the function of that gene, and so it just ends up being an interdomain linker, which I think is kind of cool. The packaging signal for SARS coronavirus 2 appears to be distributed, which means it's probably going to be something like MHV with these little, 7 nucleotide, 8 nucleotide units, but those are small enough that those would occur everywhere. And the idea is that for SARS-CoV-2, these things exist, but they're scattered. It's like the pieces of Voldemort's soul that have been scattered all around. And so they have to come together in 3 dimensions to make a shape that's going to be similar to the packaging signal that you would find in MHV and interact, and that would be what the nucleoprotein would bind in SARS-CoV-2. That's the current understanding, and yeah, we'll see how long that holds up for. So, model of the MN interaction, and it's going to be more like the top one, seems to be the more likely. So you've got the N3 domain, which is going to interact with the C-terminal domain until the packaging signal comes along. And you see the N3 domain actually had those acidic residues. And so you got 4 acidic residues. And so are those interacting where the 4 little nucleotide repeats would interact on the surface? I'm not sure. I have no idea. But that's the idea. And then the packaging signal would have a higher affinity for the nucleoprotein and vice versa. That would pop out the N3. N3 would now be free to bind to an M protein and stabilize it in the long conformation. And that would then start a chain reaction that leads all the nearby M's to start to convert. Because in the presence of N, M is converted much more efficiently to the long form. So that's the idea of how it will work. Let's look at the bioinformatics of M for a moment. So M always has 3 transmembranes here. Some of the fish coronaviruses have a signal peptide, which is like a 4th transmembrane that's going to be cut off and is going to mark the protein for transport toward the Golgi, and then if it has signals in the end that get it dragged back, like here is a conserved dibasic signal, KXX, in there, that would be something that would get it pulled back from the Golgi toward the ER, like the spike was. And so as if this is interacting with the spike or the E protein, then all of these things are getting co-transported up and down until they get to a certain size when they're able to go from flat, where they're susceptible to movement by the cell, to folding all their little tails, which is where they interact with the cell, on the inside and making a virion. That's the budding thing. And that's the point at which they're going to be detached from the cell's transport system, and they'll go down the default pathway. The default pathway being that it goes from ER to Golgi to cell surface and out. Yeah. Your packaging and assembly interactions are going to happen down here in the tail. There is a hinge region up here. That's going to be very important in conformational change during budding. And there is an ectodomain out here. This is the little part that sticks out. There's very little that is conserved in this part. But one of the few conserved things is usually there's a couple serines or threonines in there. And those would be where the glycosylation happens. All right. Each. So M is the only thing that's really needed for budding. M should be able to bud on its own, but it's much more efficient when E is there. It's even more efficient when N is there, and it is even more efficient when the genome and spike are there. So each component is helping the other ones to form the budding conformation. And the more components you get, eventually it hits a tipping point of some sort, we think, and it goes from flat to just boom, popped up like that. Now for the mechanism. The mechanism appears to be this. So you start out with the transport conformation of M. So M is sort of wide and short. We call it the compact M. It kind of looks like that. And those M's are going to have the hinge region pushed up into the membrane. So it's pushing aside lipids and it's making some space for itself in the membrane. When they interact with the N3 domain, because the N3 domain has been kicked out by an interaction with the packaging signal, then you get this interaction. And so the hinge is going to be pulled out of the membrane, and you see those two things that look like hands at the bottom of the compact? They're going to tilt down like this and become longer. So they were like this, and then they go like that. And this is the part I'm talking about, and that's what it becomes. This is now the long conformation, and there appear to be there's an ability to shift between the compact and the long conformation. So depending on what other proteins and factors it's interacting with. The long conformation makes the virion rigid, and it interacts, like these M's will interact at an angle. So there's about 6 degrees between adjacent, pairs of M's. There we go. And so you get 60 of these things forming a ring if you're just looking at a cross section of the virion. And if you look at it in 3 dimensions, tiled over the surface, there's about 1,200 of these dimers of M on an average sized virion. Larger virions are going to be less efficient in that they're going to use more M to do the same job. But if each one that's in the long conformation is putting 6 degrees of curvature in there, then that means that there aren't going to be any more long M's. It's just going to be that some of the M's you have are going to be compact still, and so they're going to be wasted M's in terms of the virus efficiency of budding. How does this work? Well, if you have 2 membranes, so imagine the red is the outer leaflet and the purple or the blue down here is the inner leaflet. What happens if these 2 are stuck together, which the 2 leaflets of a lipid bilayer are stuck together, and you shorten one of them? Now you're shortening one of them by taking out a big chunk of something that was taking up space in there. We were displacing lipid with the hinge region right there. And when the hinge region is pulled out, I've exaggerated, but this is now going to be shorter. But remember, you have 1,200 of these things all pulling out their hinge regions at the same time. That's a lot of change all in one concentrated spot. And so what happens is the membrane is going to naturally want to bend in this direction because they're still interacting with each other. The red and the blue are still parts of the same lipid bilayer, but when you're bent in an arc like that, then the short one and the longer one are now fine. They can interact with each other. That's just the way a rainbow works. The inside is shorter than the outside. All right, so we're shortening the inner leaflet, which does this, which then forces it to do that, and then M's are going to lock in that curvature by linking together, and the M's are going to lock together. The N's below them are locking together in those little spider egg configurations. The spikes are just sort of floating around. Spikes will interact with an M, but spikes are kind of far apart, and they're not really participating very much. And then E is kind of pushed out to the edge. E is just sort of lurking around the fringes. And when we get enough M's converted to make the entire thing like a sphere, that's when the E comes in, and it's going to insert amphipathic helices, we think, and that is going to cause budding, or cause scission, sorry. So this is the process. If you want to look back through it, the idea is that we go from a mix of conformations early, and then when we get all the components together, then we shift to just about entirely the long conformation. And then if we're in an endosome coming into the cell we're about to infect, then the H plus that is in there, being pumped in by vacuole or ATPase, is one of the factors that will cause the M to let go of its ends and cause it to shift back to the non-rigid compact conformation. And so that's the like the cycle. That's how it builds a rigid virion that can withstand the pressures of flying around through the air and stuff like that. And it loosens that up and it lets go of its cargo so that when it does do fusion with the spike, then it will be able to release just the free nucleoprotein. Because if it stayed stapled to M, then that wouldn't do the virus much good. It would just be stuck to the inside of whatever membrane it happened to merge with. Here's budding, and budding would just work by mathematics. So you would have a little patch imagine it in the center of a patch of M's that are all there. As they turn into the M long conformation, imagine that's the red edge spreading out. And it's just going to go, here's when it's halfway up, here's when it's about 3/4 of the way, and here's when it's basically done. So it's going to go from flat to a little dome to rising up and then sitting right back down on the membrane like that. And you can find pictures of virions doing all those things, probably a conserved mechanism for a large number of enveloped viruses. If you're wondering about the timing of this thing, which is certainly something I was wondering about, we have some clues from HIV, because it's also an enveloped virus, and it looks as though this is going to be a model for how other things work. So we have very precise information on HIV, that it takes from translation to transport to the budding site for HIV is about 8 to 9 minutes. And that's going all the way to the plasma membrane. So that's pretty good. That's a pretty good clip. From that point, it's about 16 minutes until a new virion is going to be undergo scission after budding and be released from there. If we imagine this time is similar for other viruses, and we look at all the pictures of these viruses in different positions, you can figure out that, oh, yeah, it's actually a pretty even rate. It's like 3 or 4 minutes for each of those steps to go from flat to 1/4 to half to 3/4 to tethered. And then scission would take place after that fairly quickly, within a minute or two, and then away it can go. The scission reaction, if you want to call it that, is not actually a chemical reaction. The scission action is going to be caused by E, we think. E is a transmembrane protein. Here are the transmembranes of several E's all lined up. It's got a little piece that would stick out, but there's not enough E on virions for it to be an effective way for the cell to catch virions. They would make some antibodies against this little surface bit, but not enough to do any good because there's not much E on a virion. It's thought to be something like 1 or 2 copies per entire spherical virion. But there are many more copies around the site where the virions get made, and so that's probably where they're doing their thing. So after that, there's a cysteine region, and this is going to be the region where palmitate is added. So this is like an amphipathic helix. And then you have this post-transmembrane region. And this also appears to be an amphipathic helix. And it's modeled as that, and it's been solved as that, at least in part. This is the same assay that we saw for the fusion peptide. So if you have the transmembrane region of E, and you just make that, and you throw it at these little green vesicles, you can see it doesn't really do anything. Because it may interact there, but it's only going to be stable if it's going all the way through the membrane, which means it's displacing just as much from one leaflet as the other in our sort of 2-leaflet lipid bilayer. The post-transmembrane region, from there to there, that bit is going to interact heavily with one leaflet of a vesicle, and it basically causes the vesicle to start to bend and twist to try to even out this curvature, because we're stretching out one leaflet while leaving the other one the same. So we're forcing it to do this and then bend back and then this. Yeah, it gets all wavy and weird. That is probably the membrane scission event. is inserting these things in there, in the membrane, at the spot where the tethered, budded virus is waiting to be cut loose. Influenza virus, the flu virus, has a protein called the M2 protein. That one is a known scission protein for the virus. And that one does the same thing in the same amount of time as the E protein's EPTM region. So that just tells you that, okay, whatever M2 is doing, the E protein is doing as well. And so that's how we have some idea of how this is working. Here you can see the M protein above us in the long conformation with the hands sort of tilted down like that, making a little, like a little heart, a little corona heart. Oh, look at that. Oh, wait for it. Wait till it comes back. There it is, the little heart in the middle. Yeah, corona loves you. Yeah, so that's what happens with E. And this is the other thing that E can do. This may or may not be important. We still haven't, the jury's kind of out. It seems like it should be important because it's there, but every time people do careful experiments to check if it's actually important, they come back with the answer that it doesn't seem to be important. So either we're missing something, or it does this, but it's no longer evolutionarily like needed. It's just a thing that it used to have to do and is still doing, but it's like, does it matter anymore? So there's transmembranes. E will form a pentamer with 5 little transmembranes, and then this is the region that comes after this. This is the region that's going to be inserted into the membrane and is going to be amphipathic, just sort of halfway stuck in the membrane there. And that is your E protein, and that's how it works as far as we understand it, and that's your structural protein. So that's the process all the way from expression to scission of how we make a new virion. And remember, we make the virion somewhere between the ER and the Golgi in a little vesicle. We call it the ER Golgi, intermediate compartment, but it's just like a shifting, it's like cars pulled up at a traffic light. They're not staying there. They're all going somewhere else. Yeah, traffic light's not a parking lot, and ERGSE is not a parking lot either. So once they bud into this little tiny vesicle, and you can see those in infected cells, then that vesicle carrying a virus is going to go through the Golgi and all the way up to the cell surface, and it's going to merge at the surface and release the little virion, and that's going to go free. And that's the story of coronaviruses.